Okay, so this is Miss Gray, and I'm going to go through this DNA and protein synthesis PowerPoint, and hopefully this will be much more helpful than some of the other things that we've posted throughout the course of the semester. I'll ask for your all's feedback at the end of it. So we're talking about DNA and protein synthesis. Some things that you should have gotten before you got to this point is the structure and function of DNA, and also you should have gone through some RNA and the structure of RNA. But if you didn't, we're going to go through it again. Okay, so RNA versus DNA. Here we have, on one side, we have DNA, or I'm sorry, RNA right here. And you should have already picked up that this is a single-stranded organism, or not organism, single-stranded molecule. And on this side, you should have picked up that DNA is a double-stranded molecule. I'm going to just fast forward through all the history on your notes. This should be filled in for you, and you can go back and look at it. Something, I'll just highlight some things. Watson and Crick are known as the fathers of DNA. They're the ones that discovered that DNA was a double helix. And all that means is, if we back up, DNA has two strands. Through hydrogen bonding, they twist and form this structure called the double helix. Okay, so basically what this is showing you is just, how DNA makes up chromosomes. DNA actually wraps around proteins that wrap around more proteins that condense to form chromosomes, and this is what you find in the nucleus of the cell. When the cell is dividing, we have these chromosomes that are condensed, or you might be more familiar with sister chromatids. But when the cell is not dividing, it is in the form of chromatin. And if you actually take these chromosomes and you unravel them, you'll get DNA, and one segment of DNA is a gene. So this segment right here is a gene, and some of you should be familiar with what a gene is by now, or just, a basic concept. This gene might control eye color. This gene might control skin color, and as we go on to genetics, you'll learn that eye color and skin color are actually controlled by many genes, but that could be one gene that controls for that. This is just review. DNA is made up of 3 parts. We have our phosphate group, our deoxyribose sugar, and our nitrogen base. You can have 4 different types of nitrogen bases found in DNA: adenine, thymine, guanine, and cytosine. And the way I tell my students to remember this is all tigers cry grr. Okay? Adenine is going to bind with thymine. Guanine is going to bind with cytosine. Here are the nitrogen bases. There are 2 types of nitrogen bases. We have the purines, which have double rings, and we have the pyrimidines, which have single rings. The DNA bases that we're talking about today are guanine and adenine, that are the purines or double ring, and then there are the pyrimidines, which are cytosine and thymine. uracil, you should know, is found in RNA, not DNA, but it is actually a pyrimidines. Here we have the DNA, and this is a molecule of the DNA. It doesn't quite show the double helix or the hydrogen bonding that creates that double helix, but it does show hydrogen bonding between the nitrogen bases. It shows how these bond together, and you're going to have a purine binding with the pyrimidines or pyrimidines, and adenine is going to bond with thymine, and guanine is going to bond with cytosine. You can also see how these nitrogen bases make up a rung or rungs of the ladder. This is actually a ladder shape, and the sides make up phosphate groups and sugar bases. And you kind of see how all that kind of fits together. DNA, and you should have learned this back when we talked about macromolecules. The genetic code is a sequence of DNA nucleotides in the nucleus of the cells. DNA. DNA is a double-stranded molecule. The strands are connected by complementary nucleotide pairs. Adenine bonds with thymine, and cytosine bonds with guanine, like rungs on a ladder. The ladder twists to form a double helix. DNA. During S phase and interphase, DNA replicates itself. DNA replication is a semi-conservative process. And up until this point, you should have already gone over what the semi-conservative process means, and also that DNA is actually copied during that S phase of interphase. And all we mean by semi-conservative process is that you conserve a part of the original structure in the new one. You end up with two identical strands of DNA. And if you look at this, you should have learned before that DNA helicase, an enzyme, will come and split the parent strand. It splits the parent strand, while DNA polymerase comes along on the parent strand and makes a new nucleotide sequence on there. It makes another DNA strand so that it binds complementary. So adenine bonds with thymine, guanine bonds with cytosine. DNA. So I just went over how gene is a segment of DNA that codes for a protein, which in turn is going to code for a trait, skin tone, eye color, etc. A gene is a stretch of DNA. So if we take, those sequences of DNA, adenine binding with thymine, cytosine binding with guanine, we take one strand of that DNA, and just a segment of that strand is a gene, and that gene might code for skin tone, eye color, etc. DNA contains genes, sequences of nucleotide bases. These genes code for proteins, and proteins are used to build cells and do much of the work inside the cells. So when we talk about DNA, it contains genes on it and sequences of the nucleotide bases. These genes code for proteins, and proteins are used to build cells and do much of the work inside them. RNA. Now, RNA is different, and you should have picked this up by now. RNA has, instead of the deoxyribose sugar found in, like is found in DNA, we have RNA, which has ribose sugar. And if you'll notice, the name for deoxyribose, or DNA, is deoxyribose nucleic acid, whereas for RNA, it's ribose nucleic acid, and ribose means for that ribose sugar. Nitrogen bases found in RNA adenine, uracil, guanine, and cytosine. We replace T with U, okay? So we replace that thymine with uracil. And just like adenine and thymine bind together, adenine and uracil are going to bind together in RNA. And just like in DNA, guanine and cytosine are going to bind together. Just the same in RNA. Okay, the function is to obtain information from DNA and synthesize proteins. There are three differences from DNA. One, it is single-stranded instead of double-stranded. Two, ribose instead of deoxyribose, so we have ribose sugar instead of deoxyribose sugar. And three, we have uracil instead of thymine. Okay, so there are three types of RNA. There's messenger RNA, which is mRNA, and it copies information from DNA for protein synthesis. We find what is called a codon on the RNA, and this is three base pairs that code for a single amino acid. And before we move on, you guys should have before picked up on the fact that amino acids build proteins. There are three types of RNA. Another type is transfer RNA, and it collects amino acids for protein synthesis. So what it's going to do is it's going to collect an amino acid and bring it to the messenger RNA. There's an anticodon, a sequence of three bases that are complementary base pairs to the codon in the mRNA. And then there's ribosomal RNA. It combines with proteins to form ribosomes. Amino acids. Amino acids, these are the building blocks of proteins. At least one kind of tRNA is present for each of the 20 amino acids used in protein synthesis. Okay?